other chicken, porcine, and these rodents. These are the references. This is, thank you.
Beautiful study. I was really enamored listening to you, so I was not really thinking what questions to ask. It is a very well done study. So the one thing I couldn't understand, the M31 that you described, which is the closest, but it is different from that. How did you decide on that M31? What is how is it done?
Okay, so actually, first of all, that the source of isolation is very important. So it was a human fecal contaminated, fecal contaminated soil from which it was isolated. After that, we performed some taxonomic analysis, as well as there are some parameters as I've shown in the slide. Actually, I have got one slide for that. This one. This is the whole genome. This is the whole genome data of M31, and these are the parameters which particularly defines a new species. And for an example, these are the cutoffs which is prescribed in studies, which says that whether a species is novel or not, and our values were different, and hence M31 was given as a new species. And all these values were much more higher than the Lactobacillus vaginalis, which is currently the most closest species to root rye. Yeah. And then I showed that ANI tree also, which completely depicts that how close M31 is to Lactobacillus root rye. That is why, and sir, actually when we do this comparative analysis with Lactobacillus vaginalis, we were realizing that most of the clusters which were present in Lactobacillus root rye are not present in vaginalis. That means it is a taxonomically very far away neighbor of Lactobacillus root rye. However, most of the genes, as we can see in the pan genome analysis, as we can see in this analysis, so only 55 genes are shared with vaginalis and M31, and only 55 between root rye and vaginalis. However, there are 317 genes which are shared between Lactobacillus root rye and M31. So this is actually which states that it is a very close relative of the species.
And regarding colonization, what would you comment that if root rye is not colonizing now, if you give it to humans?
So the human strains are not, yes, in fact, any of the root rye strains is not showing any colonization as far as this particular study is concerned. Like in the one I showed in this case. So there is no colonization. However, there are many trials which are going on, and they are perfectly seeing the good results of Lactobacillus root rye in humans. So what is actually happening behind that? And this was the reason that I was interested in that conversation yesterday when Dr. Dan has said that colonization is not important, because this is what is happening in case of root rye. There is no colonization, which not evidently in experiments and all those things. And in fact, the human strains does not have any gene for colonization, but still they are showing a very good results. So maybe they are working just like drugs.
So can you comment on that? What they could be doing, these probiotics?
Yeah. There could be different mechanisms for colonizations also.
No, not colonization. We know it comes and it stays. You feed them 3 times a day or something. It is going to work. So we don't care if it doesn't colonize. So it's like a drug you treat, and then during that period it will work. So what is it doing? How is it imparting
so actually, what is the actual mechanism of action? That is also not known.
Can you speculate on that?
Yeah, this is just speculations at how they are performing. In fact, that is what I'm saying, that such type of studies should be incorporated in probiotic studies. The host adaptation and whether the species is autochthonous or not. Because nobody even, I don't think so that scientific community is bothered that this particular species, that is root rye, is autochthonous or not, and whether it is going to colonize. I mean, colonization, they will study.
Let's keep the colonization part separate. We accept that it doesn't colonize.
Yeah.
Then, because you have done a wonderful work, I am pushing the envelope a little bit and asking that what could it be doing?
If it is not colonizing.
It is not colonizing, that's forgiven. We accept it, and then it is giving good results.
Yeah.
Have you looked into studies, what type of study, what type of ailment,
So
what is it doing? Is is it a uterine that is giving an impact,
Yes,
or
sir.
is it coming
Exactly.
and attaching to the
So there are some
cells?
recent studies. In the past few years, there was not any research associated with the uterine role playing a part in all these activities. However, in 2018 only, there are many studies which has come up and which says that uterine is produced by ruteri, and this shows activity against a lot many enteric pathogens, as well as it also has a good important role playing in other diseases like gastroenteritis and sepsis.
And imagine that uterine is totally useless, it has no effect. Hypothetically, I'm saying. What else could it be doing as a probiotic?
So
If it is not a secreted molecule which has antimicrobial effects,
it's not producing that.
could it be doing something else?
So maybe it is maintaining re-maintaining the dysbiosis of microbiota which is already residing in the gut, which is present in the gut. Maybe it is reinitiating, or maybe it is acting, it is aiding the other probiotics to flourish, and which is already present in our body.
So you would like to do some quorum sensing or some
Yeah,
other
exactly,
type of
exactly.
experiments to
I actually that kind of study may be requiring different interactions with the hosts, where host factors also play a role, and there will also be quorum sensing and interaction among different. This is what I think, because as this slide says, in this case, I have studied that different kind of bacterial strains, and they form colonization. They helps in colonization. So this could
Thank
be
you.
one factor of that.
Wonderful. Thank you.
Okay, thank you so much.